Hi viewers, this is J. Swami, Assistant Professor of Zoology. Today we are going to discuss the topic related to hormone action. In multicellular organisms, the cells receive a variety of signals from different sources including endocrine glands and respond appropriately. Animal cells exchange information about the concentration of ions and glucose in extracellular fluids, the interdependent metabolic activities taking place in different tissues, and the correct placement of cells during the embryonic development. In all these cases, what happens? The signal represents information that is detected by specific receptors and converted into a cellular response, which always involves a chemical process. That is, the conversion of external information, otherwise signal, into a metabolic or physiological change in cells is commonly called as a signal transduction. So there were many pioneers. They have worked on the hormone action. L.W. Sutherland Jr. was a Nobel orator. The Nobel Prize in Physiology or Medicine in 1971 was awarded to L.W. Sutherland Jr. for his discoveries concerning the mechanism of action of the hormones. So, in his statement, what he said is, when I first entered the study of hormone action some 25 years ago, there was a widespread feeling among biologists that hormone action could not be studied meaningfully in the absence of organized cell structure. However, as I reflected on the history of biochemistry, it seemed to me there was a real possibility that hormones might act at the molecular level. So he focused on the molecular level and he got Nobel Prize in 1971. Let us see some properties of hormones. These are secreted by specialized glands, carried by bloodstream, so that they are called as ductless substances, acting in remote target area, and the hormones are produced in trace amounts, causes tremendous effect. Binding to specific receptors and target organs, usually has short half-life and tightly regulated. These are the properties of the hormones. And general principles of the hormone action, there are trophic hormones, synergism, permissiveness, and antagonism or antagonistic effect. These are the general principles of hormone action. And just now we discussed that the signal transduction, the conversion of external information or signal into a metabolic or physiologic change in cells, is called as the signal transduction. There are three stages. One is the reception of the signal. This is a cell, this is the extracellular fluid, and it is the intracellular fluid cytoplasm. This is the plasma membrane. Here there is a signaling molecule. Let us see the hormone. Let us think that is a hormone. Here it is a receptor. So this signal molecule comes in contact with the receptor and it converts this signal into a physiologic or metabolic change. So thereby this is a signal transduction pathway, conversion of the signal into the response, and here there is a activation of cellular response. Three steps: reception, transduction, and the response. There are some signals to which actually the cells get respond. In general, these signals may be antigens, cell surface glycoproteins, oligosaccharides, developmental signals, extracellular matrix components, growth factors, hormones, light, mechanical touch, neurotransmitters, nutrients, odorants, pheromones, testants. These all are the signals by which the cells can respond. Here see the 3 steps signal transduction pathway STP. The steps involved in passing along the message to cause the cellular effect. These are the reception, that is detection of a signal molecule coming from outside the cell that causes change in shape of the receptor protein, and transduction convert the signal to a form that can bring about a cellular response, and response cell takes action in response to the signal molecule. These are the 3 events which are taking place in the cell signaling, basic stages of the cell signaling. And here hormones are the chemical messengers. Hormones are the chemical messengers. They act as signal molecules and carry specific message to the target cells and direct them to perform specific task. They convey the message to the cells by binding to specific receptor. So this is considered as a hormone and this is the receptors. A receptor is a protein molecule present either embedded in the cell membrane or it may be present in the cytoplasm or nucleoplasm. So if they are present in the cell surface, they are called as cell surface receptors, and if it is present inside the cell or inside the nucleus, they are known as the intracellular receptors. That is, a receptor is a protein molecule. It may be present either in the cell membrane, that is cell surface receptor, or it may be in the cytoplasm or nucleoplasm. It is known as the intracellular receptor. The hydrophilic and peptide hormones bind to the cell surface receptors, whereas the steroid hormones, which are hydrophobic in nature, they bind to the intracellular receptors. Here you can see protein that binds signals and initiate a signaling cascade. This is the cell surface receptor and this is the intracellular receptor. See here, cell membrane receptors are the integral membrane proteins that bind an extracellular signal and start a signal cascade. Whereas here, these intracellular receptors are the nuclear hormone receptor, cytoplasmic hormone receptor, otherwise nuclear hormone receptors. See here, this is the cell surface receptor and this is the intracellular receptor. Here, the signaling molecules for the cell surface receptors are the proteins, peptides, amino acids and nucleotides which cannot cross the plasma membrane. Here in the intracellular receptors you can see steroid hormones, retinoids, vitamin D, nitric oxide and the carbon monoxides are the signaling molecules. And here you can also observe the specificity of the receptors. A molecule which binds to a receptor is called as the ligand. A ligand may be a peptide or a neurotransmitter or a hormone. The receptors are specific for each hormone. For instance, for example, insulin binds to insulin receptors present on the hepatocytes, adipocytes and skeletal muscles, while the epinephrine binds to the beta adrenergic receptors on the same hepatocytes. So here you can see, here there is a messenger, ligand, otherwise a hormone. Here there are 3 cells, 1, 2, 3, cell A, cell B, cell C. Here these are the receptors. So it can fit into the suitable receptor. It interact with the chemical messenger, that is the ligand. It interact with the specific receptor on a particular cell where actually it shoots to fit into the receptor. So the cells may contain many receptors, but the specific ligand it interacts with the specific receptor, we should know. So there is a specificity of the receptors. And if you observe the types of receptors, receptor types, the receptors are broadly classified into 4 families from 1 to 4 or A to D. They are the, here you can see the type of receptor a ligand will interact with, that depends upon the nature of the ligand. Hydrophilic ligands interact with the receptors that are found on the cell surface, just now we discussed. In contrast, hydrophobic ligands can enter the cell through the lipid bilayer of the cell membrane and interact with the receptors found inside the cell, either the cytoplasmic receptors or the nucleoplasmic receptors. There are 4 families. The receptor types are categorized into 4 families. First one is the ligand-gated ion channels. Ligand-gated ion channels, G protein coupled receptors, enzyme linked receptors, and the intracellular receptors. Best example for these, the ligand-gated ion channels, they are the cholinergic nicotinic receptors, and here G protein coupled receptor GPCR, they are the alpha and beta adrenoceptors. And enzyme linked receptors, best example is the insulin receptors, and here intracellular receptor example is the steroid receptors. So these are the 4 groups: ligand gated ion channels, G protein coupled receptors, enzyme linked receptors, and the intracellular receptors. All of them, they lead to the intracellular effects. So here also you can see the ligand gated ion channels, ionotrophic receptors, GPCR, metabotrophic, and here also kinase linked and nuclear receptors and their respective examples. Here we have classified the hormonal receptors, intracellular and as well as the cell surface receptors. On the intracellular receptors, there are different kinds of hormones like the steroid hormones and the thyroid hormones act upon. So here these are the receptors. They are glucocorticoid hormone receptors, mineralocorticoid hormone receptors, androgen hormone receptors, estrogen hormone receptors, and progesterone hormone receptors. And here for the thyroid hormone, T3 and T4 receptors. And for the cell surface membrane receptors, here FSH and LH receptor, insulin hormone receptor, growth hormone receptor, oxytocin and antidiuretic hormone receptors, catecholamine hormone receptors. These all are the examples for the hormone receptors. Here location of the receptor, cell surface receptors, they interact with the proteins, peptides, catecholamines and eicosanoids, and the main effect may be generation of the second messengers which alter the activity of other molecules, usually enzymes within the cell. Whereas if you see the intracellular receptors, whether that may be cytoplasmic or nucleoplasmic, the hormones which are interacting with these intracellular receptors, mainly they are the steroid hormones, thyroid hormones which are lipid soluble, and they alter the transcriptional activity of the responsive gene. And here you can see furthermore, let us see little introduction about the 4 families of the receptors before going to the mechanism of action of the different kinds of hormones. The first family receptors, they comprise the ligand gated ion channels that are responsible for regulation of the flow of ions across the cell membrane, like the synaptic transmission. And if you see the second family, G protein coupled receptors, these receptors are comprised of a single peptide that has 7 membrane spanning regions, 7TD family, and these receptors are linked to a G protein having 3 subunits, alpha, beta and gamma subunits. Alpha subunits bind to guanosine triphosphate GTP, hence called G protein. And coming to the third group, this consists of enzyme linked receptors, that consists of those having cytosolic enzyme activity as an integral component of their structure or function. The most common enzyme linked receptors are of epidermal growth factor, platelet derived growth factor, atrial natriuretic peptide, insulin, etc., that have a tyrosine kinase activity as part of their structure. And coming to the fourth family of receptors, they are the intracellular receptors. They differ considerably from the other three. Either the receptor is entirely intracellular, and therefore the ligand must diffuse into the cell to interact with the receptors. So the best examples are the steroid hormones. They interact with these kind of receptors. Here hormones and their respective receptors. Here the peptide and hydrophilic hormones bind to cell surface receptors, and whereas steroid hormones that use the intracellular receptors. So there are two kinds of receptors, cell surface and the intracellular. Cell surface receptors, they interact with the peptide or hydrophilic hormones, whereas the steroid hormones, they use the intracellular receptors. Here peptide or hydrophilic hormones bind to cell surface receptors and stimulate the production of certain intracellular molecules called second messengers. These second messengers carry the information to the cell interior and activate certain protein or enzymes that perform specific biochemical functions. Biochemical function. That is, here there is a peptide hormone. It kept outside by binding to the receptor, but it mediates the internal activity by producing the second messenger system. We should not forget that always hormone is the first messenger. Hormone is the first messenger, whereas the hormone comes and bind with the receptor, forms the hormone receptor complex, and it stimulates the internal activity, otherwise the transduction process, where they mediate the all these activities mediated through the second messenger system, and thereby that second messenger interacts even with the gene activation. That is, the hormone may not enter into the cell, but by attaching to the receptor, it mediates all the internal metabolical activities. That is the specialty of the peptide hormones, which bind to the cell surface receptors. Here, this is the receptor here. See, if we take into consideration a particular activity like when the blood glucose level falls below the normal level, alpha cells of the islets of Langerhans, they are stimulated to release the glucagon, and the function of glucagon is to increase the blood glucose level. This is accomplished by the breakdown of the glycogen into the glucose in the liver, that is glycogen converted to glucose 1-phosphate. Glucagon released by the alpha cells of the pancreas, it is transported by the blood to the liver. The membrane of liver cells contain receptors called the beta adrenergic receptors. They are associated with the G binding proteins on the cytoplasmic phase. These called as the beta adrenergic receptors. They are bind with the G proteins on the cytoplasmic phase of the cell membrane. Hence, they are known as the G protein coupled receptors. So, these G proteins are generally the heterotrimeric proteins. G proteins are heterotrimeric proteins and contain 3 subunits: alpha, beta and gamma. Alpha, beta and gamma. When the hormone is not bound to the receptor, all the subunits of G protein are together and the alpha subunit contains a bond to the GDP and is in inactive state. When glucagon binds to the glucagon, otherwise an epinephrine, hormone binds to the beta adrenergic receptors, the G protein coupled receptor on the cell membrane of liver cell, the alpha subunit dissociate from the remaining part, dissociate from the heterotrimeric G protein, and GDP is exchanged with the GTP. GDP is exchanged with the GTP. So this GTP is equal to the ATP, and thereby the alpha subunit in turn activates the membrane bound enzyme which is called as the adenylate cyclase. Adenylate cyclase, it is present in the inner surface of the plasma membrane. This enzyme converts the ATP into cyclic AMP. ATP into cyclic AMP or CAMP, cyclic adenosine monophosphate, in the cytoplasm of the liver cells. This cyclic AMP is known as the second messenger. See here, either the epinephrine or the glucagon, this is the first messenger, and this is the receptor, and this is the GPCR, G binding protein, cell receptor, and here it is the enzyme adenylate cyclase. This is a substrate, ATP get converted to cyclic AMP. Now this is the second messenger. This is the second messenger. So this cyclic AMP called as second messenger, as the hormone which brings information is considered as the first messenger. So here the Sutherland discovered the role of cyclic AMP as a second messenger, and he was awarded Nobel Prize in Physiology or Medicine in year 1971 for his discoveries concerning the mechanisms of the action of hormones, especially epinephrine, via second messengers, namely cyclic adenosine monophosphate or cyclic AMP. So this cyclic AMP converts the inactive protein kinase A into active protein kinase A that interacts with the other metabolical activities. This cyclic AMP binds to the inactive protein kinase A that modifies the inactive protein kinase A into protein kinase A, which is active state. That phosphorylates the inactive phosphorylase kinase, and thereby this phosphorylase kinase converts phosphorylase B, which is inactive, into the active phosphorylase A. This phosphorylase breaks alpha 1,4 linkage of the glycogen from the non-reducing end by adding the phosphate group, phosphorylases, and liberates the glucose 1-phosphate. This phosphoglucomutase converts the glucose 1-phosphate to glucose 6-phosphate, and glucose 6-phosphate enters into the endoplasmic reticulum through the transporter, where the enzyme glucose 6-phosphatase removes phosphate group and liberates the glucose. The glucose leaves the endoplasmic reticulum and plasma membrane through glucose transport to enter the bloodstream and maintains the blood level. So here it is a beta adrenergic receptor, which belongs to 7TD family, 7 transmembrane enzyme family. Here the first messenger and second messenger system. This is the G protein coupled receptor. This is the largest family of the integral membrane proteins involved in the many biological processes and pathologies. 50% of all modern drugs and 25% of the top 200 best-selling drugs are estimated to target G protein coupled receptors. Transduce the signals mediated by diverse signaling molecules such as ions, peptides, lipids, and photons to induce different intracellular functions and bind their ligand and to activate different G proteins. Here we are seeing the roles of different kinds of G proteins. See here, you can also observe the 7TD family. 7 times it passes through the membrane. 1, 2, 3, 4, 5. 6 and 7 times. These are the subunits of the G binding protein alpha, beta and gamma. Whenever it is get activated, alpha subunit get detached from this and beta gamma get separated. That forms the see here alpha, beta, gamma. See here epinephrine stimulated the cyclic AMP synthesis. If you compare the resting phase and the stimulated phase, same epinephrine hormone is there. Here hormone attached. G protein is coupled. Here the alpha subunit of the G protein comes in contact with the adenyl cyclase. Thereby ATP get converted into the cyclic AMP. This is the second messenger. Hormone is the first messenger and this is the second messenger. Thereby there is a increased heart rate, dilation of skeletal muscles, blood vessels, breakdown of glycogen to glucose takes place. So he was Earl Wilbur Sutherland Jr. discovered the second messengers, won the Nobel Prize in the year of 1971 in the field of medicine. He saw that epinephrine stimulated glycogenolysis in liver cells, but epinephrine alone would not convert glycogen to glucose. He found that epinephrine had to trigger a second messenger in the form of cyclic AMP for the liver to convert glycogen to glucose. For this, he was awarded Nobel Prize in 1971. So these are the different kinds of second messengers. This is cyclic AMP. This is the normal AMP and here it is a cyclic form of phosphate, cyclic AMP. That is the change. So this ATP get converted into cyclic AMP in the presence of adenylate cyclase or adenylyl cyclase. So thereby one pyrophosphate will be get released off. So this is the cyclic AMP. So here glycogenolysis, glucagon or epinephrine, it comes in contact with the beta adrenergic receptors. This is the G binding protein system. This is adenyl cyclase. Thereby in the presence of adenyl cyclase, ATP converted into cyclic AMP. There is a conversion of inactive protein kinase into active protein kinase A, and thereby inactive phosphorylase kinase converted to phosphorylase kinase active. And here there is a phosphorylase B converted into phosphorylase A, and the glycogen converted to glucose 1-phosphate, and phosphoglucomutase convert the glucose 1-phosphate to glucose 6-phosphate, and there is a removal of the phosphate group and the glucose from the endoplasmic reticulum through the transporter reaches the blood and thereby raise the blood level, raise the glucose level in the blood. This is the glucose 6-phosphatase activity, that is transport of the glucose through the endoplasmic reticulum into the blood. So that is the mechanism of action of the peptide hormones, how they act upon the cell surface receptors. And coming to the second part of the topic, where steroid hormones use intracellular receptors. Lipid hormones such as testosterone, estrogen, progesterone do not use second messengers. They also do not activate either enzymes or proteins. Instead, they diffuse through the plasma membrane, binds to an intracellular protein present in the cytoplasm or nucleoplasm, and forms a hormone receptor complex HRC, and enters the nucleus and direct the action of genes. So that steroid hormones enter inside to the cell. They may bind to the intracellular cytoplasmic receptor or intracellular nucleoplasmic receptor, and they deal with the action of genes. See here, when you compare to the last picture of the peptide hormones, so the peptide hormone bound to the cell surface receptor, but here the steroid hormone, it diffuse through the plasma membrane and interact with the intracellular receptor, and here this is the HRC, hormone receptor complex. Hormone receptor complex, it deals with the manipulation of the gene activity. This is the cytoplasm and this is the nucleoplasm. Cytoplasm and the nucleoplasm. There are different kinds of intracellular receptors. So here the hormone receptor complex either attaches itself to the DNA or activates some transcription factors which in turn initiate transcription of specific genes and the synthesis of new proteins. For example, the administration of estrogen and progesterone causes the production of egg white albumin in female chicks, like that. See here you can see basic structure of a gene regulating intracellular receptor. These receptors are located within the cell and function in the reception of signals such as steroid hormones, vitamin D and the thyroid hormones. See here, this is a signaling molecule, signal molecule binding site, signal molecule binding domain. Here it is a DNA binding domain, and this is the inhibitor protein, and this is the transcription activating domain. So these are the intracellular receptors and their parts. Here you can see the stepwise phenomena where you can find out. Here there is a steroid hormone. It enters through the plasma membrane and enter into the cytoplasm and finally into the receptor protein which is present in the nucleoplasm. Right? There is a formation of the attachment of the steroid hormone to that of the receptor, that forms the HRC, that is hormone receptor complex. It interacts with the DNA transcription phenomena where there is a copying of mRNA based on the nitrogen based on the DNA. So here this mRNA has been formed. The mRNA from the nucleoplasm that is translocated into the cytoplasm, then thereby it get attached to the ribosomes, and there is a translation process where the tRNA molecules carry the respective amino acid to the site of the protein synthesis, and thereby that forms a new protein that deals with the metabolical activities. So like that, these steroid hormones, they interact with the intracellular receptors. So here you can observe the mechanism of action of steroid hormone and the thyroid hormone. So diffusion of the hormone through the membrane, and thereby there is a binding of a receptor to that of the hormone, then followed by binding of hormone receptor complex to the DNA, binding to the DNA, and gene activation and transcription and mRNA production, and then translation protein synthesis in the cytoplasm, and alteration of cellular structure or otherwise the activity, and that leads to the response of the target cell. Here there is involvement of the mitochondria so that there is a increased ATP production. So this is the example for the activity of the steroid hormones, otherwise thyroid hormones on the intracellular cytoplasmic receptors, otherwise the nucleoplasmic receptors. And there is a brief discussion about the second messengers. Second messengers are the molecules that relay signals received at the receptors on the cell surface from the first messengers. What is the first messenger? That is a hormone, protein hormone, otherwise a hydrophilic hormone. and that will be carried, the information from the hormone that will be carried into the interior of the cell. In addition to their job, that is as a relay molecules, second messengers serve to greatly amplify the strength of the signal. They not only carry the information from outside to the interior of the cell, but also they greatly amplify the strength of the signal. Binding of a hormone to a signal receptor at the cell surface may cause massive changes in the biochemical activities within the cells. See here, Earl Wilbur Sutherland Jr. has awarded Nobel Prize. Just now we have discussed. And the properties of the second messengers, they are short-lived intracellular signaling molecules. Elevated concentration of second messenger leads to the rapid alteration in the activity of one or more cellular enzymes. Removal or degradation of second messenger terminates the cellular response. And here there are different kinds of second messengers. There are three major classes of the second messengers. One is a minor class. So totally there are 4 classes of second messengers. Cyclic nucleotides, membrane lipid derivatives, calcium ions, and the nitric oxide, carbon monoxide is a minor one. So there are 3 major classes of second messengers. Cyclic nucleotides like the cyclic AMP, cyclic GMP, and membrane lipid derivatives like the inositol triphosphate IP3 or diacylglycerol DAG. IP3 inositol triphosphate and the DAG diacylglycerol. These are the membrane lipid derivatives and the calcium ions as we know. See here, cyclic nucleotides, membrane phospholipids and the calcium ions. Cyclic nucleotides includes cyclic AMP, cyclic GMP. Membrane phospholipids include inositol triphosphate and the diacylglycerol. The calcium ions are the calcium ions. Let us start with the cyclic AMP. Cyclic AMP, it is some of the cyclic AMP is a second messenger that is synthesized from the ATP by the action of enzymatic action of the adenylyl cyclase. Binding of the hormone to its receptor activates the G protein, which in turn activates the adenylyl cyclase, leads to the appropriate response in the cell by using the protein kinase A, pKa. It is a cyclic AMP dependent protein kinase that phosphorylates the target proteins. Otherwise, cyclic AMP binds to a protein called cyclic AMP response element binding protein, CREB. Cyclic AMP response element binding protein, and the resultant complex controls the transcription of genes. So the best example for this is cyclic AMP action on the adrenaline, glucagon and the luteinizing hormones. And coming to the cyclic GMP. Cyclic GMP is synthesized from the nucleotide GTP, guanylyl triphosphate, using the enzyme guanylyl cyclase. The cyclic GMP serves as the second messenger for atrial natriuretic peptide, nitric oxide and rods of the retina to light. So here you can observe the cyclic AMP and cyclic GMP, both of them. And if you come to the membrane lipid derivatives, peptide and protein hormones like the vasopressin, thyroid stimulating hormone, angiotensin and neurotransmitter like GABA, gamma aminobutyric acid, bind to G protein coupled receptors, GPCRs, that activate the intracellular enzyme phospholipase C, PLC, phospholipase C. It hydrolyzes phospholipids, specifically phosphatidyl inositol 4,5 biphosphate or bisphosphate. That is phospholipase C. It hydrolyzes the phospholipids, especially phosphatidyl inositol 4,5 bisphosphate, PIP2. This is commonly called as PIP2. Phosphatidyl inositol 4,5 bisphosphate, which is found in the inner layer of the plasma membrane. Hydrolysis of the PIP2 yields two products. That yields two products. One is the IP3, another one is the DAG. What is DAG actually? Diacylglycerol is the DAG that remains in the inner layer of the plasma membrane. It recruits protein kinase C, a calcium dependent kinase that phosphorylates many other proteins that bring about the changes in the cell. That is the DAG, diacylglycerol. And if you come to the IP3 is nothing but the inositol 1,4,5 triphosphate. This soluble molecule diffuses through the cytosol and binds to receptors on the endoplasmic reticulum, causing the release of calcium ions into the cytosol. The calcium ions in the cytosol triggers many types of events such as muscle contraction, release of neurotransmitters at synapses by exocytosis, secretion of hormones like insulin, activation of T cells and B cells when they bind antigen with their antigen receptors, that is T cell receptors and B cell receptors respectively, adhesion of cells to the extracellular matrix like the cadherins, desmosomes, okay, and it will be done during the process of apoptosis. So these all are related to the IP3. The calcium level. The function of second messengers, just now we discussed, that is not at all conveying the information from the cell surface to the interiors of the cell, but also amplification of the signal. Binding of few molecules of hormones to receptors produce many molecules, otherwise the products. See here, you can see calcium is used by both G protein and tyrosine kinase pathways as a second messenger. Here G protein, and here this is the calcium ions. If they are present outside, so that leads to all the muscle contraction and all those things in the cytoplasm. Here there is a tyrosine kinase, G protein. Calcium is used by both G protein and the tyrosine kinase pathways as the second messenger system. So you can explain this with the hormone and the G protein. So here also signaling molecule and the tyrosine kinase receptor. Here also the same thing, calcium colmodulin cycle. So here at last we will see the hormones that bind to the intracellular receptors are, as a conclusion, here we can see what are the hormones that bind to intracellular receptors. That is androgens, calcitriol, estrogens, glucocorticoids, mineralocorticoids, progestins, retinoic acid, and thyroid hormones like the T3 and T4, triiodothyronine, tetraiodothyronine. And second group of hormones that bind to the cell surface receptors, those which use the cyclic AMP as the second messenger. The cell surface receptor hormones are alpha 2 adrenergic catecholamines, beta adrenergic catecholamines, adrenocorticotropic hormones ACTH, antidiuretic hormone ADH, calcitonin, and chorionic gonadotropin, human corticotropin releasing hormone, follicle stimulating hormone, glucagon, lipotropin, luteinizing hormone, melanocyte stimulating hormone MSH, parathyroid hormone or parathormone PTH, somatostatin, thyroid stimulating hormone. These all are the hormones. They use cyclic AMP as a second messenger and interact with the cell surface receptors. And if the second messenger is GMP, the peptide or hormones are atrial natriuretic factor and nitric oxide. And other hormones, they use calcium or phosphatidylinositol as a second messenger, acetylcholine muscarinic receptors, adrenergic catecholamines, angiotensin 2, antidiuretic hormone, vasopressin, that is cholecystokinin, gastrin, gonadotrophin releasing hormone GnRH, oxytocin, platelet derived growth factor PDGF, substance P, thyrotrophin releasing hormone TRH. These all are using calcium or phosphatidylinositol as the second messenger system. And some other hormones which use the kinase as the second messenger: adiponectin, chorionic somatomotropin, epidermal growth factor EGF, erythropoietin, fibroblast growth factor, growth hormone, insulin-like growth factors 1 and 2, leptin, nerve growth factor, platelet growth factor, and prolactin. So these all are the hormones that use kinase system as the second messenger system. That is, there are only two groups of hormones. Some of them they use the intracellular receptors directly, and some of them they use extracellular receptors, cell surface receptors. Those which interact with the cell surface receptors, they mediate with the second messengers. So based upon the second messengers, there are different kinds of hormones and their second messenger systems. So some of them they use the cyclic AMP as a second messenger, some of them they use the cyclic GMP as a second messenger, some of them they use the calcium and phosphatidylinositol as a second messenger, and some of them they use the kinase as the second messenger system. So this is all about the receptors and their hormone action. Thank you one and all.